Hey guys, welcome to our video about cell membranes and diffusion. Today we are going to be focusing on the cell membrane, and we're going to also look at how molecules diffuse and what process occurs with that, and even through the membrane, which you're going to need these notes for Wednesday when we're doing our group activity about diffusion. So let's get started. So, if you remember, guys, that the animal cell has its cell membrane on the outside, and so this thin layer here, we're going to look at a little bit more up close in just a minute, whereas the plant cell has its cell membrane right there under, basically on the inside, we'll say, the inside of the cell wall. So this right here was our cell wall. We knew that, right? Cell wall. So this over here is our cell membrane, okay? And you usually cannot see it because it's stuck up against the cell wall. But we can look at it. We need to have pretty good magnification and staining techniques to do that. You also should understand that there's, since there's cytoplasm, all of this in the cell and all of this in the cell is cytoplasm, unless it's an organelle. It's mostly made up of water. So there's a watery environment inside the cell and a watery environment outside the cell. This is called intercellular fluid, inter meaning in between. So this is just giving you a little perspective of this, where the cell membrane is though, and what kind of fluids there are. Alright, so if we take a look at our cell membrane structure, this is a little bit more up close, looking at actual molecules here. What does it consist of? Well, it consists of these molecules called phospholipids, and there's a phosphate part of this, and then there's a lipid part of this. In fact, there's even a glycerol. We'll get into that. There's these what are called fatty acid tails to them, and why do they orient themselves that way? We're also going to get into transport proteins, and this actually, I need to draw another arrow to this. This is another transport protein right here. Okay, so this whole structure is a transport protein. Okay, this purple structure here. And then, of course, this red structure here is another type of transport protein. And then we're going to get into, later in the year, some receptor proteins that are on the surface. So these surface proteins, they're called as well, and they can either be on the outside or the inside of the cell. All right, so, and then we also need to label, last but not least, this is the cell, this basic structure of the cell membrane is what's called a phospholipid bilayer, because you can see that this is a phosphate and this is a phosphate. We'll talk about why they orient themselves in just a second, but that makes a bilayer, two layers, here and here. Okay. All right, so we have these proteins here, and basically this right here, this is important, we're going to have to understand that, is a channel. So, or an opening. So make sure you label that a channel or opening in the cell membrane, which that protein is structured to have. It's almost like a tunnel for molecules to pass in and out of the cell. Okay, we'll talk about that later. All right, so that's the same thing I just showed you. This is basically what a phospholipid looks like more closely. This has a phosphate, what's also called a polar part of its phosphate structure. It's also got a glycerol molecule, which is part of the fat or the lipid, and that glycerol allows these fatty acids to attach to it. So basically it has these fatty acid, and I almost say tails to them. That's not really that accurate, but fatty acid tails and a phosphate head to it. Okay, so why do they orient themselves? Oh, I'm sorry. Why they orient themselves to that is that the head of this is actually what's called hydrophilic. Hydrophilic means water-loving or attracted to water, or then there's a hydrophobic. You think of phobic, think of fearing. It's trying to get away from water. All right, so it's repelled by water. Okay, so you may want to pause that just to get that caught up. And so what does this look like? Well, we have water outside the cell, and we have water inside the cell. And what happens is these phospholipids, they orient themselves where the phosphates are attracted to the water. Water is polar, means it has a charge on one side. It basically sets up two charges. Oxygen kind of has a negative charge to it on it, and these phosphates have a positive charge, so they're going to be attracted to that negative part of the water. So they're attracted to it, whereas the lipid tails, just think of oil and water, don't mix. These are going to try to get away from it or repelled from it. And the same thing happens to these phosphates. So these are attracted to the water, whereas the lipids are repelled, and so they're trying to get away from each other. So these tails are trying to get away from the water, and these tails are trying to get away from the water, and that's where they come together in a phospholipid bilayer. Alright, so that's enough about the cell membrane. What we need to think about is diffusion. You're going to really need this for Wednesday. So diffusion, if we look at it, and think of some sort of sugar or salt. And I like to think of Gatorade, that's used in our example on Wednesday. If you had the Gatorade mix, right, and you put a scoop of Gatorade into water, well, the Gatorade molecules are going to be very close together. Once you put them in water, they're going to start to diffuse or dissolve and then diffuse. And we also know that molecules in a liquid or even a gas are going to move randomly. So this molecule might be moving in this direction, this molecule might be moving there, this molecule moves there. Okay, so, and they even collide with each other. So these two molecules might collide, and when they do that, they may bounce off and go into opposite directions. So we know that when we put that sugar into the water, right where we put it in is probably going to be where we have it most concentrated. The molecules are really packed close at first. But after about 5 minutes or so, we would see that the molecules spread out. And what does that look like? Well, you can see that there is more space between these molecules. They're taking up, that scoop of Gatorade is taking up the whole jug of water after a while. And we can see now that the molecules are in a lower, considered to be a lower concentration because they're spread out. So that's what we have to think about with high concentration versus low concentration. Is high concentration, the molecules are really packed close together. But low concentration is they spread out. And this is because of the random movements of molecules bouncing off each other and moving in all different directions. So diffusion, you got to get this part down at the bottom, is the movement of molecules from high to low concentration with no extra energy expended by the cell. This happens randomly from molecules just moving and bouncing off of each other, and at room temperature certainly molecules are moving pretty rapidly, so no energy is needed by our cell for this process. So diffusion through a membrane we have to look at is when we're comparing two different sides, the inside of the membrane, inside of the cell to the outside of the cell, or vice versa. We might go outside to inside. So what is that? Remember, we have to look at that, and you know what the inside of the cell looks like. We got cytoplasm, which is mostly water, but it has those dissolved sugars and salts and other nutrients and waste products and all that other stuff. And then outside the cell, we have that intercellular fluid surrounded by a watery environment, but it also has other salts and sugars and other things that are dissolved in there in that intercellular fluid as well. So sometimes we like to just take a piece of that membrane just to compare it, and we just look at it up a little bit more close. And what does it look like? It looks like this. This is the cell membrane. Okay, so we call this a semi-permeable membrane, and let me just get this out of the way. Semi-permeable, and we're going to talk about that. That's the whole idea of Wednesday's lesson, is what is a semi-permeable membrane. You may want to look that up. But remember, environment 1, environment 2, this might be inside the cell and outside the cell, or vice versa. Sorry about that. And so just keep in mind, semi-permeable membrane, we're also talking about the cell membrane. That's what we're referring to. So this cell membrane is our example right now of the semi-permeable membrane we're going to be talking about on Wednesday. There are other semi-permeable membranes, but for now we'll just keep an eye on that cell membrane, and we'll talk about semi-permeable membranes as well. Just another couple of things that you should know about. Keep this in mind, help you. A solute, that's a chemical word for the substance being dissolved. So this is like your sugar or your salt that you put into the water, and it breaks up into small, fine molecules that you can't even see, and it's being dissolved. The solvent is what's doing the dissolving. And in a lot of cases, especially with our body, water is the most common solvent. You think about all the things that can dissolve into water. Not everything dissolves in water, but there are quite a few things that are important nutrients and waste that do dissolve into water for us. And then we also have to keep in mind concentration. We talked about this a little bit. Concentration is basically how much solute you dissolved into the solution. Okay, so those three terms we need to understand when it comes to our types of solutions. A couple different types of solutions we'll talk about is hypertonic. Hyper means greater. Okay, so hyper is greater, so that basically means the cell is sitting in a concentration of, the solution has a greater concentration of solute. So like it might be in salt water. If you put the cell in the salt water, that'd be a hypertonic solution you just put that cell into. You can kind of see that the salt inside the cell is definitely low concentration, and the salt outside the cell is in a high concentration. So we have to think about what's going on with that. The hypotonic solutions, those are the solution outside is lower concentration, has a lower concentration of solute. So it could be salt, lower concentration of salt, but a higher amount inside the cell. And isotonic. Isotonic is basically where both sides of the membrane there is an equal amount of solute. There also could be an equal amount of solvent in this case too. Basically an equal concentration of solute and solvent. That's a more important thing, a way of saying it. And that they're packed the same way, okay, around there. And they're going to be randomly moving in and out. And this is what we talk about with that process of dynamic equilibrium. We're going to do another vodcast on that one as well. But for right now, just understand your different types of solutions. All right, I think that basically comes to the end of our notes for now. You might want to watch a couple of the animations on osmosis that I put on the website and diffusion. That might help you as well. I'll put those links on Wednesday and provide those to you as well, hopefully. And that'll be it. So these are your notes, your vodcast. I think we're up to vodcast 15, cell membrane and diffusion. And I hope to see you in class. All right, talk to you later. Bye.